Our last group of tests is the immunological methods. The immunological methods utilize serology. So serology is testing of serum. Serum contains antibodies. So antibodies are the proteins. They're Y-shaped proteins. These are made by the human immune system. There's billions of different antibodies in your body. Each antibody reacts to a specific antigen, and the antigen will attach here to the arms of the antibody. So each antibody is specific to the antigen it'll attach to. Whenever somebody is exposed to an antigen, whether it be a virus, bacteria, even an allergen, whatever it is, the immune system will create these antibodies. Specifically, it's the B cells. B cells will be activated. B cells, once they're activated against an antigen, will convert into plasma cells. Plasma cells are actually the antibody producing cells. And some of the B cells will form memory B cells, so that if the person's ever exposed again to the same antigen, the cells will activate very quickly to plasma cells and make these antibodies. Immunological methods all utilize these antibody-antigen interactions. It can go either way. Maybe we're testing an unknown antibody to see if it reacts with a known antigen, or maybe we take an unknown pathogen from the body, we don't know what it is. These methods are highly specific because, like I mentioned, antibodies are very specific to the antigen they attach to. So although there are times we might get false positives or false negatives, normally we see that these interactions are highly specific, so they're reliable in that way. And they're also very sensitive, meaning even if we only have small amounts of antibodies in the serum, we will still see a reaction, because even small amounts of antibodies will react to antigens. So we can detect even when somebody is newly diagnosed with a disease, or maybe at one point in their life they had the disease, but it's been a long time, so the body hasn't made antibodies against it for a while. So it's very sensitive in that way too. So these methods can not only be used to detect if somebody is currently infected with a disease, it could also be used to show if somebody is maybe newly infected, or if they've ever been infected at some point in their life, then they'll probably have antibodies against the pathogen. So like I said, serological testing can go in two different directions. Either we could take a patient's serum, and we don't know if this patient has antibodies to a certain disease. So I'll give you an example, the coronavirus, okay? Has this patient been infected before with the coronavirus? We don't know. So we take the patient's serum, then we take the known antigen. So that means we take antigens that we've isolated from coronavirus. So we know these are known coronavirus antigens. And then we mix them together. And what we're looking for is to see if the patient does have antibodies against the coronavirus. Then, like I said, the antibodies will bind to the antigen. And you'll usually see some sort of reaction, either clumping. We can measure it using clumping. We could do color. We could measure it via fluorescence. So it depends on the test we're doing. But in some way, we need to be able to visualize if the antibody and the antigen are reacting with each other. So this is how we can actually diagnose or see if somebody at one point was infected with a certain pathogen. If they do have antibodies, then we'll see this reaction. It can go the other way around as well. Let's say that somebody comes in with a really strange food poisoning and we don't know what it is. We don't know what the pathogen is. We can try to isolate the pathogen from the patient. We can grow it up on a petri dish. So now we have, in this case, an unknown antigen, whereas over here in this case we had an unknown antibody. It can go either way. So in this case, we have this unknown antigen, this unknown pathogen that we grew on the petri dish. We maybe suspect it's something, like we'll say, oh, maybe it's listeria, who knows? So then we take known antibodies against what we think it could be, and we mix them together. And again, we're looking for some sort of interaction between the antibodies and antigens. Let me give you like a real life example of this. So in this example, a patient has some sort of bacterial meningitis. And as you know, there's many bacteria that could cause meningitis. It could be strep B, Haemophilus influenza, strep pneumonia. It could be Neisseria meningitidis, strain ACY or W135, or it could be Neisseria meningitidis B or E. So in this case, we have the known antigens for all of these different bacteria, and they're on these kind of cards, and then we place the patient's unknown antibody. We don't know what the patient has, so we place the patient's serum onto the card, and we're looking for that clumping. And of course, you see the clumping here in sample 4, indicating the meningitis that this patient has is being caused by strain ACY or W135. And then the opposite, the alternative, we could take an actual bacterial sample from the patient. So in this case, we have an unknown antigen, some sort of unknown pathogen from the patient. Again, put it on the card, and then we could put known antibodies, so a known anti-serum against Neisseria meningitidis. And again, we're looking for that clumping here. So when you see that clumping, this indicates a positive result. If you don't see that clumping, it indicates a negative result. So like I mentioned, all of these serological tests, we need a way to visualize if the antibody and the antigen are reacting with each other, because antibodies and antigens are so tiny, you can't actually see them. So you need a way to know how the reaction turns out. So one of the ways we know is we can do an agglutination test. So an agglutination test, it's basically, this means clumping and big clumping. So for example, if you had a whole virus or a whole bacteria, okay, the whole cell, and antibodies present, the antibodies could bind. Remember, there's 2 arms of the antibody. So one arm could bind to one pathogen, the other arm could bind to the other cell. And same thing over here, and same thing over here. So what you end up getting is this huge clump of whole cells with antibodies. Or similarly, if you had a virus, like in this picture, these antibodies will attach to multiple viruses and they cause kind of this massive clump. So whenever you're doing an agglutination test, it's the antibody basically reacting or attaching to the entire cell or the whole virus, the whole agent, causing these massive clumps. And if you're doing this reaction like in a test tube, in a liquid test tube, these clumps are going to be so big that they're going to all fall to the bottom of the tube. So you're going to basically see these clumping, these agglutinated cells that are going to fall to the bottom of the tube. So that's what an agglutination test does, basically. Many times when we're doing these kind of tests, we'll do serial dilutions like we've been doing in lab. And the reason that they do these serial dilutions is because they want to measure the titer of antibodies that are in the serum. So the titer basically tells you the quantity of antibodies that are present in the serum. This doesn't always tell you everything, but it can give you some idea of basically when the patient was infected. If a patient has a lot of antibodies to a specific antigen, it means that they've recently or are currently have that antigen in their body because their body's immune response is reacting to it currently. A lot of times we could tell basically the timing of the infection or how the body's responding by looking at this titer, the quantity of antibody. Now, similar to agglutination, we could also measure if antibodies and antigens are interacting using a precipitation test. So in a precipitation test, you're not looking for the whole cell or the whole virus interacting with the antibody. Instead, you're just looking at like a very small antigen. So something tiny that's interacting with the antibody. And similarly, if there's a lot of these tiny antigens, you're still going to get these kind of clumps, but they're not those massive clumps like you saw in the agglutination test. Instead, it's what we call precipitation. So precipitation is more like a cloudiness. So you wouldn't see these big clumps, but you might see this kind of cloudiness if you were to do it in a liquid test tube. Most precipitation tests we actually do in an agar petri dish. We use a little bit less agar so that the antibody and the antigen can kind of diffuse around in the agar. So how this test works is we might have these little wells. We might stab these little wells into the agar, and then we would add the antigens and the antibodies. So for example, in this second picture, we put the antigen here right in the middle, so we inject some antigen in there. Then we would put the antibodies around the antigen. So this was a control. That's a positive control. And what you see in the positive control are these little bands. So you know that the antigen and the antibodies are hooking up together. So that precipitation would appear as bands. And you can see in test serum 2, there's a band. So this individuals positive, but test serum 1, there's no band, so it's negative. This picture is showing you like a real life example. So 1 and 4 are supposed to be the positive controls here, and you see the 2 bands. And you could tell patient 2 and 3 were both positive because they have bands, but patient 6 and 5 were negative because there's no bands there. So an example of a precipitation test that we actually do in real life is the VDRL test. This stands for the Venereal Disease Research Lab Test. This is used to detect if somebody has antibodies for syphilis. So basically we would put the syphilis antigen here, and then we would be looking for these bands to see if the person has syphilis antibodies, indicating they are infected or have been infected at some point. Our next test to talk about is the Western blot. The Western blot is one of the most specific and sensitive tests out there. It's used usually as a secondary test, like for example for HIV or for some other diseases, right? The first test used is almost always an ELISA, which we'll talk about later. So if an ELISA test comes back positive, then to confirm the positive result, we would use a Western blot. And the reason we don't use the Western blot first is because it's a little bit time consuming and it's not like the most easy test to do. You need like a really skilled laboratory technician to do this. So it's used to verify a positive result. So here's how Western blot works. Remember in the last video we talked about gel electrophoresis? Well, we're still doing gel electrophoresis in this case. But instead of doing it with DNA, we're doing it with proteins. So for example, we would take, if we're talking about HIV, we would take this HIV virus and we would extract all the protein from it. And then we would put all the protein in this gel and the gel would run. And remember, smaller proteins are going to run faster. So what you would get is all of these HIV proteins that are going to show up on the gel. So that's from a known HIV antigen. We know it's there. Now after this, all of these HIV antigens are now on the gel. Now we're going to add the patient's serum, the patient's antibodies. If the patient does have antibodies that interact with HIV antigens, they'll kind of react with one another, and usually we use a color change. So you'll see that these bands are going to turn blue. So over here in this picture shows you an example. This first lane is all of the HIV proteins with positive results. So this is what it would look like if all of the antibodies were present to bind to antigens. You can see all of these names are different HIV proteins like GP160, GP120, and they're all on this gel spaced out by their size. Then what you would do, this is an example showing a patient on day 0. So this would be basically if a patient was negative for HIV. You'd see there's no blue bands other than this one here, which is a positive control. So this indicates that the patient was negative for HIV antibodies. This is what it would look like 9 days after contracting HIV. So 9 days post-infection, the person has now started creating some antibodies to some of the HIV antigens. And then as you can see, as time goes on, there's more and more blue that shows up. Another blue band shows up here. Another blue band shows up here. So over time, more and more antibodies are going to be created by the immune system against these HIV antigens. So the Western blot not only tells you if the patient has antibodies against these antigens, it also can give you an idea of when they were exposed. If they have very few antibodies, it might be a recent infection. Whereas if they have all of the HIV antibodies, it could mean that they've had a long-term infection.